we are looking at stem cells and cell differentiation. Now, we've already kind of covered off on the mitosis part, which is quite in depth, but our focus this lesson is to learn about stem cells in particular and what makes them unique. All right, let's look at unicellular versus multicellular organisms. It sounds pretty self-explanatory, but our unicellular organisms are very simple. They can carry out the 7 functions of life in one cell. Some of them move, some of them are stationary. They have different features of them that make them able to move, and their cell size is limited by surface area to volume ratio, which we'll cover off on a bit later. Some live in colonies, so they work together. They're not fused, but they work together, and we're talking yeast, bacterium, amoeba, paramecium. If we're talking multicellular organisms, we're talking a one big single fused mass of cells, and they are cooperative cells here. We often talk about emergent properties, which means the sum of the parts is greater than just all the little bits, right? So we're getting a whole bunch of properties because all these cells are fused together. Our levels of organizations within an organism, we're talking very simple atoms, squished together to give molecules. We're going to get macromolecules, these huge giant molecules that will form parts of our cell. We get to the cellular level, they combine to form tissues, which combine to form organs, which combine to form systems, and that will give, all those multiple systems will give us our entire organism. But before a cell can be part of tissue or organ system, it must have some certain distinguishing features that actually allow it to do its specialized job. And a precursor cell is known as a stem cell. It's unspecialized, so it's kind of like a generic or default cell. It has the potential to be any kind of cell in the body. And stem cells can do 2 things. They can replicate to make more stem cells, or they can differentiate, which means they can turn into other more specialized cells. Now, the genetic code, or our DNA, is essentially in charge of determining what pathway our cells take and how they differentiate, or if they do. And it depends on which genes, which are segments of our DNA, which ones are switched on and off, as to what will determine what proteins, what features those cells are going to have, and therefore what job they do. So cells in some tissues or some organs may have similar jobs, they might have slightly different jobs. For example, our red blood cells actually remove their nucleus. They build up huge supplies of a protein called hemoglobin so that they can efficiently carry oxygen and carbon dioxide around our body. And these look, similar but different to an ovum, which is an egg, and they have all these nutrients in their cytoplasm to start off embryonic development. They're specialized membranes so that they can kind of shut down as soon as one sperm enters or burrows in, essentially. We've got muscle cells. They're really bundled fibers close together so they can contract and relax, and they contain so many mitochondria. So you can see how all these different cells have different features so that they can do their specialized job. Plant cells contain those photosynthetic chloroplasts. Pancreatic tissue is really cool. It has, you can see, 2 distinct kinds of cells there. Some of them are making digestive enzymes, some of them are making hormones. We've got intestinal cells, and they're arranged in these large big folds so they can increase their surface area to get more absorption of nutrients. You have sperm cells that have not very much cytoplasm. They're pretty streamlined because they have to swim really fast. They've got a lot of mitochondria to make energy so they can actually swim to that egg. They have enzymes in their head so they can burrow through that egg. And you've got quite a lot of different xylem and phloem tissue in plants organized in such a way that it allows for, water to get from the roots of a really tall tree right to the top. But before they can actually differentiate to become those kinds of cells, we need, stem cells sitting there to be the precursor to them, and they have to then differentiate to become different types. And along that differentiation line, we have different kinds. If we start off, the totipotent cells, they're the first cells formed once a zygote is created, so a sperm meets an egg, and they are embryonic stem cells, and they're only produced at this stage, like the really, really early stage. They have the potential to become any type of cell, which is how that fetal development can occur. Now, pluripotent stem cells are also embryonic. They have huge potential to become almost any type, except one, which is placental cells, but that's not very common. And they eventually become more and more specialized so that once that infant is born, they are limited to what cell type they can actually become. So you can see that they start to become multipotent stem cells, and they're able to produce certain types. So for example, you might say a precursor blood cell, and there's lots of different kinds of blood cells, or nerve cells, something like that. Unipotent stem cells are that very last stage, and they're able to specialize to become only one type of cell, which is their own, right? So that's that reproducing of the same kind of cell. For example, your skin cells have stem cells which can self-renew to create just new stem cells, but they're still really handy to have because they're maintaining those tissues. Same thing like with germline cells like sperm and eggs. So both multipotent and unipotent stem cells are mainly functioning to repair and maintain the tissue, and they're considered adult stem cells as opposed to the totipotent and pluripotent, which come out at the embryonic stage, which are embryonic stem cells. This is a lovely flowchart from Oxford that is breaking down that flow of differentiation, essentially, and there's plenty of nice color-coded charts to show similar things as well. Now, stem cells can be used to generate healthy cells, to replace disease cells. They can be used to model how diseases progress. They can test new treatments, whatever it is. But obviously, there's a lot of regulation on the collection and use of them, and that can limit the progress using them, I suppose, in research. And they can also be very controversial to people. So embryos can be discarded by IVF clinics if they're not required, and some feel that they could be used for scientific purposes, but obviously their use isn't considered ethical to some groups in our society. And while using adult stem cells obviously comes with fewer ethical issues, they're really hard to find within the body, and they have less potential for differentiation. There are umbilical cord stem cells, that can be stored once a baby is born. And these are a bit less controversial. So there's this constant push and pull between the stem cells' potential for scientific discovery and practical and ethical ways of obtaining them. Now, we're constantly trying to find new advancements in finding stem cells in the body so that they can be used, and scientists have also been able to reverse the differentiation process in certain adult stem cells, and that allows them to become toti or pluripotential cells, so they don't have to go finding embryonic stem cells, which, you know, that's a pretty handy thing to have. There is a lot in this about stem cells, so I understand that these have been heavy topics, but please make sure you are having a look specifically at what the syllabus is asking you to know.